Hi everyone, it's Jobs here, and in today's video I'm going to cover the metabolic pathway known as glycogenesis and the diseases associated with abnormalities within the pathway. Here, glyco refers to sugar. Glycogen is the form in which sugar is stored inside of the body. And genesis simply means formation. Therefore, glycogenesis is the formation of glycogen from the buildup of single sugar units. We already discussed the importance of glucose as a source of energy in the previous video involving glycolysis. However, not all the glucose taken from food will be immediately utilized for energy production. In fact, some will be stored for later necessity in the form of glycogen. So where is glucose stored in our body? The major storing sites include liver and skeletal muscles. Minor storing sites instead include kidneys and intestine. Now let's understand how glucose is stored as glycogen through glycogenesis. The very first step involves phosphorylation. Here, the glucose molecule entering the cell gets immediately phosphorylated. The phosphate group is stripped off from ATP and added to the 6th carbon of the glucose molecule, producing glucose 6-phosphate. The reaction is catalyzed by glucokinase if it occurs in the liver. Instead, it will be catalyzed by hexokinase if the reaction occurs in the brain or muscle tissue. If you have watched my previous video over glycolysis, you already know that this phosphorylation step is crucial to trap the glucose molecule inside of the cell. The second step involves the relocation of the phosphate group. The reaction is catalyzed by phosphoglucomutase. Here, the phosphate moves from carbon 6 to carbon 1, producing glucose 1-phosphate. The next process involves the activation of the glucose molecule. Here, uridine triphosphate is utilized to produce the activated form of glucose, known as uridine diphosphate glucose. The byproduct of this reaction is inorganic phosphate, which can be utilized in the steps of glycolysis for energy production. The reaction is catalyzed by pyrophosphorylase. The fourth step involves the addition of glucose to glycogen residues. This addition is catalyzed by glycogen synthase, where glucose will be bonded to glycogen residues by glycosidic bonds between carbon 1 of glucose to carbon 4 of glycogen. This extends the chain of glucose molecules being stored. The arrow shown in the figure represent the glycosidic bonds. Next, we will look to an additional step which is fundamental for the efficient storage of glycogen. This reaction is catalyzed by branching enzymes. As you saw in the previous step, glucose molecules attach themselves to glycogen through glycosidic bond between carbon 1 and 4, producing a linear structure. However, this limits the storage ability of cells. Therefore, branching enzymes removes a section in the linear structure and relocates it as the branch through the formation of glycosidic bond between carbon 1 and 6, as you can see on the right hand side. To visualize the importance of this step, imagine a tree inside of a building. If the tree was allowed to grow only upwards, it will be limited by the ceiling and growth will stop. However, if the tree can branch off, growth will still continue. And the same happens with glycogen. Branching allows more space to be covered, facilitating storage. Before we continue to explore the next part of the video, I would like to remind you to leave a like to help me reach out more people that will benefit from this type of content. Also, do subscribe. This would allow me to grow a community interested in science. Now let's explore the diseases associated with glycogen and its storage. The two major diseases related to glycogen are von Gurk disease and Anderson's disease. Von Gurk disease, or glycogen storage disease type 1, is caused by the deficiency of the enzyme known as glucose 6-phosphatase. This enzyme catalyzes the removal of the phosphate group from glucose 6-phosphate. This conversion is required to allow glucose to leave the cell when the body needs immediate energy, or when it has to restore blood glucose level through the breakdown of glycogen stored. However, when there is the deficiency of the phosphate, the conversion cannot happen and glucose 6-phosphate cannot leave as it is. As previously mentioned, the phosphorylation of glucose into glucose 6-phosphate at the very beginning of glycogen formation is there to trap the glucose inside of the cell. The symptoms associated with this disease include frequent sweating, weakness and convulsions, apnea, which is the temporary cessation of breathing during sleep. This results in enlarged kidney, enlarged intestines, as well as hypoglycemia and hepatomegaly, which means the enlargement of the liver. This occurs because glucose cannot leave and build-up of glycogen continues. Another disorder associated with glycogen storage disease is Anderson disease, known as type 4 glycogen storage disease. This is caused by a genetic defect in the branching enzyme, known as glycosyltransferase. The defect causes the inability of the enzyme to branch the glycogen structure by forming 1,6-glycosidic bonds. This results in very long and linear glycogen structures. As glycogen is not stored efficiently, the muscle tissues and liver present an unusual shape, and this causes an autoimmune response by the immune system, which target muscle tissues and liver as they are the major storing sites of glycogen. This results in poor muscle tone and liver damage, which causes cirrhosis. I hope you enjoyed this video. If you did, please leave a thumbs up and subscribe to this channel. Thanks for watching, and I will see you in the next video.